Adoptive cell transfer therapy is an approach where we take lymphocytes or immune system cells from patients, take them to the laboratory, activate them in some way, and give them back to patients. It's been an area that has been mostly an academic exercise for the last 20 years or so, where very few sites were able to do these kind of therapies. But now we have clear evidence that it works. And now it's becoming something that's going to more academic sites, and it's also being picked up by several companies. The approaches that are being used for adoptive cell transfer therapy are twofold. One of them is taking lymphocytes from tumors, what we call tumor-infiltrating lymphocytes, or TIL therapy. And those lymphocytes are expanded from tumor biopsies and then given back to patients. The other approach is to take blood lymphocytes, which are taken to the laboratory. They're genetically manipulated to give them receptors to target specifically the cancer, and then they're given back. Out of this second approach, there's two sub-approaches. One of them is using the natural receptors, or the T-cell receptors, or TCRs. And the other one is using artificial receptors, which are called chimeric antigen receptors, or CARs. They're all different approaches. The TIL therapy has the benefit of already knowing that there's some T cells in the tumor that have tumor specificity. They're taken out and they're given back. The problem is, if there's no TILs in the tumor, then the therapy will not be expanded. The approaches that are based on taking peripheral blood lymphocytes and genetically modifying them can be done in all of the patients and can be better controlled. They can be done within a short period of time, where a large number of these TIL lymphocytes can be genetically modified in a batch and then given back to patients. The problems are that then we have to use viral vectors to be able to genetically modify these, which increases the scrutiny of this type of therapy. The TIL therapy has been most advanced in patients with melanoma. It can also be used in some other cancers, but there's less evidence that that sufficient numbers can be generated routinely. But recently there was a report in cervical cancer targeting HPV viruses. The T-cell receptor engineering has been done with targeting melanosomal antigens in melanoma, but also cancer testis antigens like NY-ESO. And that seems to have the most used in a particular type of sarcoma called synovial sarcoma. The approaches based on chimeric antigen receptors are finding their best use in lymphoid malignancies, in particular targeting CD19 in both B-cell leukemias and B-cell lymphomas. These studies are going from small trials in academic centers that were more proof of principle to now being multi-center studies, some of them randomized or some of them pivotal single-arm studies. There's an ongoing randomized trial of TIL therapy versus standard of care therapy in patients with melanoma that's being conducted in Europe, centered mostly in the Netherlands and in the UK. And there's studies of CD19 chimeric antigen receptors that are multi-center being opened in several parts of the United States. This is a completely different type of therapy for cancer. It's based on making personalized cell therapies for every patient. So every batch is a new batch. It comes from a patient and goes back to a patient. So that requires a different mindset of how to develop drugs for oncology. But since we know that they work, because there's clear evidence that the adoptive cell transfer of large numbers of T lymphocytes that are antigen-specific induces regressions in cancers like melanoma, sarcoma, or leukemias and lymphomas, then these approaches, the limitations of these approaches are being overcome rapidly and move forward in the clinic. There's been a lot of excitement with immunotherapies for cancer. And in many instances, what has given benefit in the clinic has been taking away breaks to an immune system that was there ready to attack the cancer. But we're seeing that the benefit should be taken to more patients. And since what induces the tumor response is an army of lymphocytes that are cancer-specific, in some cases they're not there. And now we know we can genetically create them by taking lymphocytes outside the body, giving them the receptors to target the cancer. It's like putting a GPS on the immune system and then giving that back. And that's giving responses in several cancers. I think the near future, this form of therapy is going to be more broadly applicable. There's many limitations. It's a personalized cell therapy. But I think that the field is moving forward quickly into addressing those limitations and making active therapies.